to understand, translate, and intervene with food allergies. And we have established a unique collaborative network of leading scientists focused on bringing us one step closer to a cure. In a short period of time, FASI has changed the landscape of food allergy research, and we are very excited for the road ahead. Through our seminar series, Our Journey to a Cure, we will take you along for a journey through FASI's endeavor to create a world without food allergies. Join us for our monthly seminars where we will spotlight critical science on the path to developing transformative therapies. And today I am thrilled to be joined by Steve Liberals to discuss how the brain listens to signals from allergens to sickness, which is a key area of research for FAST investigators trying to unravel the biology of food allergies. So Steve, thank you for joining us today. Before we start, I just want to remind the audience that we will take questions from our Q&A box at the end of the discussion, and I hope people can engage with us today. Steve is a professor in the cell biology department at Harvard Medical School and also a Howard Hughes Medical Institute investigator in addition to FAST investigator. And Steve has been doing incredible work in understanding allergen sensing pathways and the communications between the gut and the brain, the enteric nervous system, and the brain. And we are very, very excited for everything that Steve has been doing. So Steve, welcome. Thank you for making the time and joining us today.
Yeah, thank you, Carlos. Happy to be here.
So Steve, it is very exciting to see someone, you and I share at least two things in common, our passion for food allergies and for food allergy research, and that we're both chemists by training, but obviously you're a lot smarter than I am, and somehow you found your way into doing something very unique and very exciting in terms of looking at the neuroimmune connections in the body, especially how the brain communicates with different parts of the body and how that affects food allergies. So if you don't mind sharing, I think our audience would really enjoy hearing from you a little bit about your background, how you got to do what you're doing, which is amazing and fascinating work.
Oh, well, thank you again, Carlos. Yeah, so it was a long and circuitous route to food allergy. I actually don't suffer from too many allergies. I may have a scallops allergy, although I'm not entirely sure. I haven't really wanted to poke that and test that to really find out. But I was trained as a chemist, as you mentioned, and I worked with Stuart Schreiber at Harvard. And then for my postdoc, I became very interested in understanding how the brain works. I think some of the great questions of our time are understanding how the brain generates our complex behaviors, moods, emotions. And so I did a postdoc with Linda Buck, studying sensory neuroscience. Linda was famous for discovering the odorant receptors, and I was actually very lucky when I was there. She actually won the Nobel Prize when I was there, so that was really great to see. And when I was a postdoc, we surprisingly found a second family of olfactory receptors that was distinct from the canonical ones. These were called trace amine-associated receptors, or TARS. And so when I began my lab, I went with Linda to Seattle, and then I came back to Harvard to start my lab. I began working on olfactory mechanisms, looking at what some of these receptors might detect and how they work to generate perceptions and behaviors. And over time, my research interests evolved, became very interested in body-brain signaling. So the brain not only receives input from the outside world, but also receives input from the body. And some of those inputs are what drive our most basic emotions, hunger, thirst, even moods like racing heart or butterflies in the stomach can cause anxiety. And so much less is understood about how the brain receives these various inputs. And so that became a major focus of my lab now.
Exciting. No, I mean, those are all very important areas that are associated with food allergies, and really one of the reasons why we've been very excited about everything you've been building, the tools, the technologies to allow the field to learn more about this and those communications. So So Steve, if you don't mind explaining to people, actually, yeah, just thinking a little bit more about that on a high level, how the brain communicates and connects with other parts of the body when, to create a response, that would be, I think, would be very helpful.
Yeah, so I should have said that, of the many signals that the body sends up to the brain, signals from the respiratory, signals about breathing, from the cardiovascular system about things like blood pressure, and from the gut, things like nutrients, but also the immune system is in constant communication with the brain. And so we've been very interested in that in the context of allergy, in the context of pathogen responses, what are the diversity of pathways for neuroimmune crosstalk? And so the body can send signals to the brain through two main pathways, right? They can send hormones and cytokines that travel through the bloodstream, and then there are specialized sites in the brain that can receive these hormones, and that lack of what's called a blood-brain barrier, and they can receive these hormones in blood. And second, there are neurons that act as direct relays from the body to the brain. And so we've been very interested in both pathways, but today I'll highlight some of our particular work on the vagus nerve. So the vagus nerve, this is a major body-brain communication pathway that controls breathing, it controls heart rate, and has this really beautiful anatomy. So there are sensory neurons of the vagus nerve that go from the body up to the brain, and there are also motor neurons that go in the opposite direction. And the sensory neurons, the cell bodies of these neurons, they live in these ganglia at the base of the neck, and from there, they send projections throughout the body to the heart, the lung, the stomach, the intestine, and these are some of the longest neurons in your body. So they interface with internal organs, and the ascending axons also cross through the skull and enter the brain, where they can evoke perceptions, moods, and change physiology. And so vagal sensory neurons detect a tremendous diversity of stimuli in the body, and we're only at the beginning of understanding all the amazing things it does.
Fascinating. I always find it fascinating how, the signals can travel distances like they are in such a fast way and to be very effective at communicating. Um, Steve, one of the things every time I see you present your data and your science, I'm blown away by all the technologies and tools you and your lab have been building to dissect this field and all the questions that remain unanswered in this field. It's just very exciting. So I'm wondering if you can share a little bit more about how you're studying this, what are the technologies you guys have been developing, and the tools that are very unique and innovative to be able to answer some of these critical communications and questions.
Yeah, thanks. So we spent a long time now developing tools to study the vagus nerve, and the first thing we wanted to do is understand its complexity, right? So we've used a variety of approaches to understand how many different types of neurons there are in the vagus nerve. And when you think of other sensory systems in your eye, you have things called rods and cones. Melanopsin is a third type of cell. In the taste system, there are different taste cells that mediate 5 different types of tastes. So salty, sour, sweet, bitter, umami, which is savory. Here we see dozens of different neuron types. And it's really exciting. There's one neuron type innervate the lung and control breathing. There's another go to the stomach and control feeding. And there are more neuron types than known reflexes. And so knowing that all of these different neuron types exist in the vagus nerve, we spent a good deal of effort to build genetic tools that we can control different types of neurons. So we can either activate them artificially, and see what happens to the physiology of the mouse. We can eliminate them and see what happens. We can put fluorescent proteins in that allow us to visualize the neurons and see where they go in the body and where they go in the brain. And we can also report on the activity, use methods to report on the activity of these different neurons. So these are, there's this toolbox that exists in mouse genetics called Cre recombinase. So we can put this gene in different types of vagal sensory neurons, and that gives us a genetic handle to understand what these neurons do. So that's been a major toolkit in the lab. And the types of things we can do is, as I mentioned, we can map where the neurons go in the body. So these are the GPR65 neurons that I mentioned. When we only label them and no other type of vagal sensory neuron, we see these beautiful neuron terminals in the intestine and in the stomach. And in the intestine, this is a cross section of the duodenum, the very proximal part of the small intestine. And you see these finger-like structures that sort of point into the lumen of the intestine. This is where the food will be. And these are called villi. And the GPR65 neurons have these beautiful fibers that innervate intestinal villi. And we think they're communicating with cells in the intestine that send the signal up to the brain. We also use another technique called optogenetics, where we can turn on different neurons using light. So there's a protein developed by Karl Deseroff called channel rhodopsin that you can express in different neuron types. And when you shine light on the neurons start firing. And if we put this protein in all different types of vagal sensory neurons, you can powerfully control autonomic physiology with light. And so this is in the mouse. And if we activate all the vagal sensory neurons, you have these tremendous changes. The animals stop breathing, their heart rate plummets, their blood pressure plummets, they get changes in gut motility and gastric pressure. If you change the anesthesia, you start seeing other reflexes emerge. The animals will start just swallowing repetitively or gasping. And so there's an amazing amount of physiology that's controlled by this nerve. But when we do the same thing, but we're activating just small groups of neurons, we can get highly selective results, highly selective changes in physiology. So these are the two Cree lines I showed a couple of slides ago. When we activate one of them, we get powerful changes on breathing. When we activate another one, we get changes in gut physiology. And so each of these neurons engages neural circuits that control these whole body physiological systems with high selectivity. And so we're trying to understand the different roles of these neurons in body-brain communication. And one last tool that we have is that we can image the responses of these neurons. And so we put a reporter that glows every time the neuron fires. And in this experiment, we're just shining light, we're just electrically stimulating the vagus nerve trunk. And every time we provide a pulse of electrical stimulation, we can see the neurons firing. And so we can do the same thing, but instead of providing electrical stimulation, we can then stretch the stomach and see that some neurons fire, or change mechanical properties in the airways and see other neurons fire. And moving forward, we've been interested in understanding if we add allergens or we add different immune challenges, do we see different neurons firing, and which neurons are responding and how do they do that?
It is fascinating to see this, especially having the exquisite control that you have with optical manipulation to be able to control those pathways. In an era where just knocking in and knocking out something, it's necessary to understand, but the exquisite selectivity that you have and control with optical tools really provides a great approach to be able to dissect some of these questions. Getting to, I think, more specifically to nausea, now that you bring this up, how do you think about, nausea is a common feature in food allergies and allergic reactions. How do you think, from your neuroimmune research, how do you see nausea and sickness, and how do they communicate, and what do they have in common?
Right. So under the large umbrella of what the vagus nerve does and other aspects of body-brain physiology do is they communicate with the immune system. And there are a variety of different challenges that the immune system reports to the brain. So one is pathogen responses. And one, allergen will also evoke a neuronally orchestrated response of anaphylaxis that includes changes in cardiorespiratory tone and also nausea-related behaviors. And so we're very interested at the basic level to understand the number of different pathways that the immune system communicates with the brain and how those orchestrate different aspects of physiology. And I just highlight two examples here where we've been interested in neuroimmune crosstalk, nausea, and respiratory infections. And so nausea is unpleasant sensation of visceral malaise, and it's part of a gut-to-brain pathway that is evoked by ingested toxins, by gut pathogens, but also by allergens, drugs that treat cancer or diabetes, as well as pregnancy and disorienting motion. And so we've been studying brain circuits that are part of the gut-brain axis that trigger nausea behavior. In separate studies, we've been interested in respiratory infections and how respiratory infections induce sickness behavior. Now, everyone knows what it feels like to be sick. Unfortunately, most people are sick several times a year. And sickness is also a neuronally orchestrated state with common symptoms such as fever, lethargy, decreases in appetite. But in addition to these common symptoms, there are also symptoms that are tailored to the sites of infection. So as I mentioned, a gut infection will induce nausea, while a respiratory infection may make you cough. And so the first question is, how does the brain become aware that there is an infection in the body? And classical studies raised models where cytokines or bioactive molecules traveling in the bloodstream can inform the brain by either specialized structures that lack a blood-brain barrier or crossing into the brain to tell the brain that there's a pathogen there. But the idea of chemicals that travel in the bloodstream, that doesn't enable a spatial, the brain won't know where the infection is located in the body. And so we thought that peripheral neurons that directly innervate the body might be able to provide that sort of spatial information. And so we were very interested in understanding the contributions of both neurons and hormones and cytokines in the generation of sickness behavior. So one set of experiments we did is we began to look at respiratory virus infections in mice. And so, influenza infects almost 1 billion people worldwide, kills close to 500,000 people worldwide. And of course, respiratory viruses in general inflict substantial human suffering, and the COVID pandemic, recent COVID pandemic, broadly disrupted human society. So if you infect mice with influenza, they display some of the same behavioral symptoms that we do. They eat less, they move less, they drink less, and they often don't survive. So what happens if you prevent the brain from becoming aware that there's an infection in the body? And so to get at this, our hook was that we leveraged the powerful anti-sickness effects of non-steroidal anti-inflammatory drugs, chemicals like ibuprofen, found in Advil or aspirin. And the way these molecules work is they block the biosynthesis of a key hormone, a key cytokine called a prostaglandin. And so we first showed that if you give mice ibuprofen, it makes them feel better. So they eat more, they move more, they drink more, and they even survive a little better, which was initially surprising to us. And so we use this as a hook to understand, to identify the pathways that are relevant. And what we ultimately found is that there was a population of neurons that innervate the airways in a region called the nasal pharynx and provide a direct signal from the nasal pharynx up to the brain. And so we can eliminate the receptor for these prostaglandin molecules only in these neurons and nowhere else in the body. And that phenocopies what happens when you give ibuprofen to the animal. And so we think these neurons are activated by prostaglandins. They send a signal up to the brain that results in sickness behavior. Now, what's really exciting, I think, is you might notice that the beneficial effects of ibuprofen are mostly early during the infection. And so when you infect the animals with flu, the virus is really prevalent in the upper airways and the nasal pharynx initially during an infection. And then as the infection progresses, the virus moves into the lungs. And so we think there's a second pathway that kicks in that doesn't depend on these neurons and doesn't depend on prostaglandin signaling. And so, for example, if you treat with ibuprofen, you have this really protective effect early. And this suggested to us that maybe there was a second pathway that was kicking in, a body-to-brain communication pathway that was kicking in later during the infection. And it turns out there is, and we can block this pathway using a different antagonist. And so when we do this, we see that the effects on sickness behavior are even better. The animals feel even better, and even though the virus is still in their body, and they actually survive more, again, which was, again, surprising to us. So we're very excited by the idea. We think this pathway comes from the lungs to the brain, and that there are multiple types of neurons that can report from body to brain on the presence of an immune challenge. And we're interested now in understanding when, across all these different neuron types, when are they activated, by what sorts of chemicals are they activated, and can we block them and control either allergen or pathogen responses in a way that's beneficial to the person.
No, it is interesting to see. I mean, you're mapping all of this for nausea, sickness. How about if you extend it now to anaphylaxis, which is something obviously very concerning for food allergies? Is it the same, similar pathways? Is it different neurons? Can you tell us a little bit more about that?
Yeah. So so the question is, how are there different neural circuits that respond to allergens compared to pathogens? That's exactly what we're exploring with the support of FASI. And not only from the sensory pathway from the body to the brain, but also the motor response is very different, right? And so in anaphylaxis, you get very characteristic cardiorespiratory changes that don't occur during a pathogen response, right? So we're thinking about understanding how the brain can then communicate back to the body, generally to control the immune system, and also in the context of anaphylaxis. And so classically, there are two routes which the brain communicates to the body through the autonomic nervous system. One is the parasympathetic, and one is the sympathetic nervous systems. And these mediate either the rest and digest response or the so-called fight or flight response. And what we found is when we look at the neuron types that comprise each of these systems, that there actually is a heterogeneous group. There are about 10 different types of parasympathetic neurons and also a multitude of different types of sympathetic neurons. And so we're very interested in the idea that maybe it's not just one program, but there are many types of parasympathetic and many types of sympathetic programs. And so we're interested in moving forward in understanding which of these might be relevant in anaphylaxis and other immune challenges, which of these provide feedback control over the immune system. And if we can redefine how the parasympathetic and sympathetic nervous system works through the lens of cell types, we'd be very excited. And just to highlight the importance of this, Kevin Tracy has done really groundbreaking work where he's shown that stimulating vagal nerve can impact diseases like rheumatoid arthritis through what is proposed an anti-inflammatory reflex from the vagus back to the immune system. Now, there are challenges with getting, the approach he uses is just to stimulate the entire vagal nerve trunk. And this has some challenges. First, it's invasive. Second, requires these complex surgical manipulations. And what they do is they turn the activity of the vagus nerve up until you start getting a sore throat, and then they dial it back. And so that's not, first off, it's not super selective. You're hitting all the sensory and motor fibers potentially. And second, you might be missing neurons that aren't activated at the level of the sore throat stimulation. And so if you can imagine finding the cell types that are actually relevant for neuroimmune crosstalk, and you can develop pharmacological approaches to target them with selectivity, you might have some of the same therapeutic benefit of vagal nerve stimulation, but now without some of the side effects. And so I think there's tremendous therapeutic opportunity here.
That point really resonates. I'm just thinking when, some patients undergo treatment like oral immunotherapy, right? The aversion and the reactions that they have that really connects to everything you're talking about. If you're able to tease out which are the neurons and the mechanisms that are involved with that, maybe people would be able to have a way to at least try to control those reactions, and hopefully we'll have more effective therapies in the future. This is amazing, Steve. I mean, I know you're building a lot of tools, a lot of understanding that would be instrumental to tackle the questions that need to be answered in food allergy research. We are out of time, but I want to leave some time. I want to have at least leave some time, 5 minutes for questions and answers. And I already have at least 2 questions in the queue here. So before we go to questions and answers, maybe I just want to remind people of our seminar series. Steve, I really thank you so much. Every time I talk to you, I learn something new. So thank you for spending the time. I know we get asked how can people help? And you can donate, host events, follow us on social media, and join our next webinar. You can use this QR code to get more information. Our next webinar is going to be very exciting, and we would actually have our scientific founder Ruslan Mesitov that will be talking about how the gut-brain communications initiate protective behaviors in food allergy. So slightly different topic, similar communications, but or similar areas of communication, but slightly different topic than what we're covering today, more in the protective behaviors that Ruslan and his team has been working on, and actually collaborating also with Steve, of course. So with that, I would open the discussion for questions. I think one of the questions, Steve, that's coming up is, what's next? What are the next questions that remain unanswered, that with everything you're building, that it's important for food allergies that you're thinking about and your team are working on?
Yeah, so what we really would love to do is define the rule book of neuroimmune crosstalk. So we know there are a couple of pathways, a couple of neurons that are relevant. there's prostaglandins play a particular role in neuroimmune crosstalk. Serotonin from the gut contributes to nausea based on the powerful effects of HTR3A antagonists. GDF15 is another gut-derived signal that can induce nausea-related behaviors. So how many pathways are there? When are they engaged across different types of immune challenges? And what sorts of signaling factors do they utilize? We know that inhibiting prostaglandins has revolutionized medicine, right, in terms of pain and sickness relief. And so what happens if you understand the entire playbook by which that underlies all these responses? So that's the major feature, I think.
There's a lot to be built, but really necessary if we're going to be able to have a big impact on allergies. There's several questions here. Let me probably go with the first one. Were there any implications on recovery from infection when these neurons were ablated?
Right. So, if you read a review paper on sickness behavior, the first line is that sickness behavior is an evolutionarily um, uh, derived program that helps conserve energy to fight off the infection. So why do the animals actually survive better if you eliminate the program? And so we were surprised initially by that. And we think there are a couple of possible reasons. So the immune system has this very delicate balance between, fighting off the infection, but also causing damage to the tissues. And the reason people die from influenza is due to the hyperactive immune response, more so than from damage directly to the pathogen. And so I think if you would take other pathogens, is something we're looking at now, if you took bacterial pathogens, who's probably provided the primary driver across evolution, you might have the opposite phenotype. The animals would do worse in the context of a bacterial infection. And so Rousson actually had a beautiful study that if you force feed mice during a flu infection, it's beneficial. But if you force feed mice during a bacterial infection, it's harmful. And so I think that's consistent with that. So the changes in behavior could be due, but the changes in response to the infection could be due to changes in behavior, but also could be due to changes eliciting motor program from the brain back to the immune system that also influences cytokine production and the immune response as well.
Thank you, Steve. I want to be respectful of people's time, and we only have 1 minute left. So unfortunately, we can get through all the great questions we've been getting. We'll try to answer via email. But Steve, thank you for taking time out of your very, very busy schedule.
Yeah, thank you, Carlos, for the invitation. And
thank you again. And we'll definitely be very excited for you to be part of FASI and have the opportunity to talk to you and learn from you. So, and thanks for everyone for tuning in. Stay tuned for the next seminar next month.
Thanks.